Good afternoon, everyone. Welcome to the Office of the Chief Scientist Science Seminar Series. For those of you who don't know me, my name is Mark Edwards, and I'm your host for today's seminar. On behalf of the entire OCS team, welcome to today's event. In the spirit of reconciliation, I would like to respectfully acknowledge that today we are broadcasting throughout Alberta and beyond on the traditional lands and gathering places for Treaty 6, 4, 7, 8, and Treaty 10, and the homeland of the Métis people. Wherever you find yourselves today, I encourage you to recognize and acknowledge the shared relationship that First Nations, Inuit, and Métis have with the land upon which we all live, work, and play. Now, in keeping with the tradition of gathering, these science seminars provide an opportunity for us to get together, to exchange knowledge on interesting science happening across Alberta in an open learning and sharing environment. The hope is that through our dialogue today, we are all able to improve our awareness and understanding and put this knowledge into practice to support evidence-informed decision-making wherever that occurs. Now, to help maintain our call quality today, your video and audio have been disabled. You can enter your questions and comments at any time during or after the presentation via the chat. After the presentation has concluded, I will read out the questions in the order that they were received. Today, we are welcoming Dr. Louie Lopez and Dr. Ave Dersch, presenting on Reconstructing a Century of Change in Lake Ecosystems, Insights from Environmental DNA and Indigenous Knowledge. Dr. Lopez is a Liber Aero postdoctoral fellow at the University of Victoria. He is part of an Indigenous community-based monitoring work plan under the Oil Sands Monitoring Program, leveraging the use of environmental DNA, eDNA, from lake sediment cores. He is part of several global initiatives for making international standards for eDNA methods and building a framework related to FAIR principles when it comes to sharing eDNA data. And Dr. Dersch has 20 years of experience working with First Nations communities in the fields of traditional land and resource use, archaeology, and community-based monitoring of traditional lands and resources. She currently co-chairs Alberta's Oil Sands Monitoring Indigenous Community-Based Monitoring Advisory Committee as a representative for First Nations in the region. She is an adjunct professor in the Department of Anthropology at the University of Alberta. And now over to you, Dr. Dersch, to begin.
Thanks. Si Ave Dersch Hushier Thlotila Alisna. That just means my name's Ave Dersch and I work for Chipewyan Prairie First Nation. Happy to be here. I'm going to start with the first part of the presentation and hand it over to my colleague, and he's going to switch the slides for us. We can go to the next slide, please. And I'm going to talk a little bit about Chipewyan Prairie First Nation. It's a Dene Sutlene community. It's often known as Janvier. It is in northeastern Alberta generally, but it's about 120 kilometers south of Fort McMurray in the Athabasca Oil Sands region. And I get a lot of support on this work through youth monitors and land users and elders, and of course from Chief and Council and the Industry Relations Corporation. That is where I work out of. Chipewyan Prairie is a pretty small community, and they actually didn't have a road connecting them to places like Lac La Biche and Fort McMurray until 1987. They live on Bond Lake, which is Janvier, and where they have a reserve, and they also have reserve lands on Cowper Lake, which we're going to talk about today, as well as Winifred Lake. Next slide, please. So this is the team here. You can see I'm bottom left-hand corner, and you can see Louie there as well, and Dr. Karen Helbig. She's would be sort of like Louie's boss, and Keegan, a lot of you guys probably know him, and he's helped us out a lot, and Mark. Paul has moved on from the government of Alberta and works in the States now, but he was a big player in this work too. Next slide, please. So the foundation of a lot of this work is two-eyed seeing approach, and that refers to learning to see from one eye with the strengths of Indigenous ways of knowing, and from the other eye with strengths of Western ways of knowing, and to use both of these eyes together. And when I think about this, I always reflect on when I was young, going to the University of Calgary. I had a part-time job. I worked at the Calgary Zoo. And there was this place, a sanctuary for injured owls. And these owls, they only had one eye. They'd been hit by vehicles or collided with other things. And we always explain to the kids they are in this sanctuary because they can't hunt anymore. They've got one eye. So when they're trying to dive to kill a mouse or something, they don't have any depth perception, and they're going to run into a branch or the ground or something. And they're not very effective hunters anymore. And we used to do this exercise where we'd have kids close one eye and try to put their fingers together and then have both eyes. And we talked about stereoscopic binocular vision in the depth perception. And that's what I always think about here. When we use these different ways of knowing and we get that crossover, that's where we can get this depth of understanding. So I always think back to the owls that can only see out of one eye. And something I also just want to remind everybody that we're certainly using this two-eyed approach. We were never using some of the Western science DNA approaches to confirm or validate the Indigenous knowledge. In this case, we were not sure this approach would even work. So the Indigenous knowledge came with a question and a change that they'd observed, and then we wanted to see if this technology would work or not. In many cases, we see more of this complementary work, and Indigenous knowledge is a really great way of detecting change. We sometimes monitoring takes a long time. You got to figure out baseline. It takes a number of years. Indigenous knowledge, the elders can often say, we've seen this change. And then maybe we choose to use Western science, maybe for investigation of cause or something. But I just wanted to reiterate, this wasn't about verifying the Indigenous knowledge at all. All right, next slide, please. All right, so there's a academic Polly in 1995. He introduced the concept of shifting baseline syndrome, where our perception of what is natural shifted towards a more degraded system. So therefore, it's become difficult to evaluate the current state of an ecosystem or make future projections without knowing the history, magnitude, and drivers of past change. Historical reference points are thus critical to measure and interpret long-term changes and to set meaningful targets for management, restoration, and recovery. So another academic, Jardine, in 2019, he said, hey, Indigenous knowledge, that's the remedy to this. That's the remedy to shifting baseline syndrome. And he asserted that Indigenous knowledge has the potential to advance a narrative of past environmental conditions as appropriate baseline against which to judge current state. So that's what we're doing here. We're using Indigenous knowledge to help define a pre-development baseline in this case. Next slide, please. Okay, so Cowper Lake, that's the picture in the bottom of the slide here. That's Councillor Arnold Black. And I'd heard stories from elders and land users, and they talked about when their grandparents would eat fish, white fish at Cowper Lake, and how they disappeared in the mid-20th century after a hill collapsed into a lake. It was kind of a catastrophic event. But when I would talk to people, maybe someone like Keegan or Mark, and they'd look in the records, they'd say, oh, well, no, our Western science doesn't say there's whitefish there. And we looked and, oh, well, that's the 1980s. That's the first time that they started to do some of their surveys in Cowper Lake. It had been many years since the whitefish had disappeared. So I think that's a really good example of this pre-development baseline and how Indigenous knowledge was able to tell us the past conditions of the whitefish there. Okay, next slide, please. So in this slide, I'm going to just talk a little bit about methods. That's Cowper Lake. You can see where it's located, and that's our makeshift sort of outdoor lab. So the elder took us, it was Paul and I in this case, to the deepest part of the lake, and Paul's really good at doing cores. So we used a gravity core and we collect a sediment core. We came back to the shore of the lake. We had our little lab set up, and we took little cookies out of these cores, and we were really careful to reduce contamination of the samples by focusing on the inside of the core because we thought there could be some smearing and contamination on the outsides. Some of the subsamples went to University de Quebec for dating through radioisotope analysis, and then subsamples would be the ones that that Louie worked on for the sediment DNA analysis. So I think that sets the stage for Louie's portion, and I'll hand it over to him. Thanks.
Hello, everyone. So before starting my section for this presentation, I'd like to express my gratitude to being able to work with the Chipewyan Prairie First Nations for the trust on handling and also processing this sediment core samples. And then I'd like to reiterate Ava's points earlier, wherein as a science inclined person and as a scientist in training, we don't come into this project wherein we are thinking to verify the Indigenous knowledge. Our thinking is to see potential of using sedimentary DNA or eDNA as an approach on how we can augment and explore synergistic opportunities on building comprehensive story regarding the history of the lake. So for this project, the main objective is to explore the possibility of using sedimentary DNA from the lake. Basically, sedimentary DNA is a subgroup of environmental DNA, which is basically DNA released by the organism and suspended to any environmental matrix. What's good with sedimentary DNA in general is that it is a good natural archive of biological information, because each layer of the sediment that settles at the bottom of the lake base is basically containing thousands of DNA released by the organism and then preserved in between sediment particles. So we're trying to explore this approach and see if we can reconstruct historical change in biodiversity within the lake history. So specifically, Ave and Paul collected the sediment core from Copper Lake in 2023. Basically, for a sediment core, the topmost layer of the sediment core is basically the youngest sediment section, while the bottommost layer or the bottommost section of the sediment core is basically older because they settled earlier. Sedimentary DNA coring can be an effective and a faster way for providing retrospective analysis of the changes in biodiversity data, especially if you're using DNA as a proxy, because there's a lot of advances when it comes to technology for detecting environmental DNA. So for this particular work plan under the OSM, we implemented two approaches for detecting sedimentary DNA. The first one is species targeted. So species targeted in a sense that it works very similar with COVID testing. So it's basically using almost the same machines and the mechanism on how to detect a DNA or a nucleic acid from any sample. We're using qPCR, or you can also use digital PCR or digital droplet PCR that would specifically detect target DNA region for a particular species. So that's why it's called species targeted, because it's very specific to a known species target. On the other hand, the other method that we implemented for this work plan is that we used DNA metabarcoding. So in comparison to species targeted, it's more leaning towards the community-based analysis. So instead of targeting a particular region of a DNA for a species, we are using conserved or shared DNA sequences among different taxa so that we can amplify those DNA target region and then see an overview on the community assembly or community compositions of the species present from or detected from the DNA pool. So the first figure that I'll be presenting is basically the result from the chronological analysis or dating of the sediment core. So the orange bar represents the estimates when it comes to the age of each sediment core section. So the oldest section of the sediment core, which is basically the bottommost section or the bottommost depth of the sediment core, is basically dated 1905. And then the youngest section of the sediment core was basically dated 2019. So we have roughly a century worth of DNA data from this sediment core collected from Copper Lake. With the help of Colin Cook and his student, we basically modeled the sedimentation rate from the sediment sections. And then as you can notice, there is a sudden spike of sedimentation rate around 1940s. I think this is around 1948 based on this model. To give you an idea, sedimentation rate is a good proxy for understanding potential input of terrestrial nutrients going to the water column. So looking at this spike in sedimentation rate during this time period, there's a potential increase in terrestrial input of loading coming from the peripheral side of the lake going to the water column during this time. And then next is that we use targeted species eDNA detection according to the Indigenous knowledge circulating around the Chipewyan Prairie First Nations. There's a lake whitefish. There's lake whitefish present within Copper Lake during 1950s or mid 20th century, but suddenly it disappeared. So based on our targeted analysis of eDNA from the sediment core, we basically detected presence of Cisco DNA from 1920 up until to 1948. So that's the approximate time period based on the chronological analysis of the sediment core section. And then after 1948, we're no longer detecting any DNA from Cisco. And just to give you background when it comes to taxonomy of whitefish within Alberta, and I think most of you would probably know, Cisco and lake whitefish are closely related taxa. So we're having difficulty delineating this two species. Just looking at the morphology, fish biologists would tell you that it might be hard or it can be a bit difficult to identify these two species. And when we try to examine the genetic sequence when it comes to the mitochondrial genome, they are around 97 to 98% closely or very similar. And also there are literature indicating that there's potential possibility of hybridization between these two species if they are both present within a particular lake environment. So, but based on DNA data from the sediment core, we're detecting Cisco DNA from this time period. And this is where we try to integrate the knowledge from the community, particularly the Chipewyan Prairie First Nations, based on the information provided by community leaders and knowledge holders. As Ave mentioned, there has been documentation of natural landslide on the eastern portion of the lake on the around 1940s to 1950s, and this could possibly explain the increase in sedimentation rate noticed on the sediment analysis. And then there's also other different changes in land use around the area. For example, the industrial development in the Athabasca oil sands region. There's also some forestry development in the area, and also in particular the oil sands development in the area, which according to Ave, it's around 30 kilometers away from the lake. So just looking at this figure, so there's particular critical period within the lake history that we want to unravel and to explore. First is the potential disappearance, or explaining what are the possible reasons for the disappearance of the whitefish species within the lake, and also is it really the landslide that caused this disappearance of the whitefish within the lake. So this is the data from the first year of analysis under this work plan, and we use species targeted because we know the different fish species that's of interest for the Chipewyan Prairie community. Just to follow up with the analysis, we've also conducted chemistry analysis. So within the different sections of the sediment core, we've noticed that there's a sudden spike in the organic matter within the 15 to 20 centimeter depth of the sediment core. And when we dated it, it's roughly very close to the dotted line presented in the figure, which represents the time frame when the landslide happened. So it kind of supplements the idea of like there's a possibility of a potential landslide within the area. and it augments the narrative coming from the community. Another thing that we did from the sediment chemistry is that we've also analyzed the sulfur isotope. That's a good proxy for a potential increase in primary productivity. And just to give you background, primary productivity could give us an insight on an overview of potential algal bloom because there's an increase in photosynthetic activity within the lake. So based on the orange dotted line, that's the sulfur proxy. So that indicates potential increase in primary productivity before the landslide happened. Another proxy that we've measured based on sediment chemistry are the alkaline earth metals, such as calcium, strontium, and barium. So based on the spike in the strontium, this is a good proxy for potential idea on the microbial productivity within the area. There's a spike same time with the primary productivity prior to the landslide. And it's also visible on the accumulating increase in sedimentation rate on the previous figure that I've shown you. So this basically gives us an idea on the past scenarios within the water column. So it gives you an idea on what are the existing water quality measured during the time frame. There is an increase in primary productivity, possibility of algal bloom. There is an increase in microbial activity. If you're at the bottom portion of the lake, increase in microbial productivity can deplete the oxygen content. That's make it less inhabitable for some fish species. And this gives us an idea that these are the potential scenarios that have caused the disappearance of the fish populations in Copper Lake. And this is basically based on another knowledge stream, which is basically coming from the chemistry folks of this work plan providing this data. And we just want to see how the data will complement with each other and how can we provide a more comprehensive narrative of what happened within the lake during this time period. And trying to expand the eDNA analysis that we've collected based on so that it will not only be limited on the fish, and we can have an overview on the ecosystem function based on the different trophic level within the lake. We also conducted an eDNA metabarcoding that will cover detection of photoautotrophs and mixotrophs. Basically, these are the photosynthetic organisms capable of doing photosynthesis within the lake and contributing to the primary productivity. We can also detect some presence of parasites if there are potential parasites that might have caused the disappearance of the whitefish populations during this time period. And there are also the possible detections of the consumers. For example, different fish species, mammalian DNA, human DNA, and other consumer or higher trophic level species. So this is the data from the DNA metabarcoding, and the dotted red line represents the time period wherein the estimated landslide according to it's around 1950s, 1940s period for the potential landslide according to the Indigenous knowledge. What I'd like to focus on this figure is that you can see that before 1950s, I'll call it pre-1950s in the Copper Lake, it's highly dominated. The DNA pool that we are detecting is highly dominated by Embryophyceae. So basically what are Embryophyceae? So these are terrestrial land plants. So before 1950s, the DNA pool that we are detecting from the sediment sections are basically highly dominated by pollens of terrestrial land plants. So I'd just like to clarify that these are insights. This does not correspond to the coverage or vegetation around the lake. So it just gives us an idea that there could be a higher vegetation cover during 1950 or before 1950s. That's why there's abundant of pollen settling at the bottom of the lake. So it's high. The DNA pool is highly dominated with terrestrial land plant. There's also the time period during 1938 wherein there's a sudden shift wherein the DNA pool is basically dominated by chlorophyceae, which are green algae or green phytoplankton capable of doing photosynthesis. And shifting on the post 1950s, I'd like to point out that it basically shifted by being terrestrial land plant dominated. There's still in their DNA pool. We're still detecting pollens from terrestrial land plant, but basically we're starting to notice increase in DNA coming from phytoplankton species, such as Basilia ruffici, basically green phytoplankton, diatoms, and dinoflagellates. These are microscopic photosynthetic organisms capable of increasing primary productivity within the lake. So there's a shift from terrestrial land plant DNA pool dominated into a phytoplankton dominated DNA pool. So it gives you an idea that there's a potential increase in nutrients within the water column that allows this phytoplankton to thrive after 1950s. And focusing on the another marker, which is CO1, it's more focused on amplifying consumers or animal DNA. So the red bars represents the fish DNA. So this approach also confirmed our analysis from the species targeted. So we're still detecting the Cisco, and most of the DNA for those fish are basically Cisco DNA during 1919 up until to 1943. And then we're also seeing a lot of insect DNA indicated by the purple bar. And then we're also detecting a lot of mammals DNA represented by the mammalia. So for the mammal DNA before 1950s, we're detecting like bison DNA. There's also pig DNA. There's a lot of human DNA. And then when I presented this data to Ave, what I'm thinking is that are there potential activity of like animal domestication within close to the lakes, or is there a possibility of settlement of Chipewyan Prairie First Nations close to the lake, because it could explain the for example the pig DNA, or it's also possible that it could be coming from the feral pig population. And then Ave confirmed, and also other biologists confirmed, that bison could be like uh naturally or has native distribution within the area. And then post 1950s, we're detecting a more diverse DNA pool coming from different animals that includes invertebrates and also insects and different animal DNA. So it's kind of hard to infer based on this figure, but we tried to examine a difference in terms of the community composition or community assembly before 1950s and after 1950s, just to assess potential impact of the landslide in the DNA pool. And then based on the phytoplankton marker, we're basically seeing a significant difference before 1950s and 1950s and after 1950s. So that's indicated with the 18S marker. And for the animal marker, which is the CO1 marker, it's kind of hard to tell that there's a significant difference between the pool before 1950s and after 1950s. So we also tried to expand the analysis based on the idea that the landslide could affect the diversity of the different DNA pool that we collected. So we also indicated the on the left side, which is the red bar or the pink bar, it's basically the pre-1950s, and the blue bar indicates the diversity after 1950s. So overall, we've noticed that there's a decline in diversity of DNA pool that we've detected after the landslide, which kind of makes sense because it's after 1950s, we've noticed that phytoplankton started to dominate the DNA pool, and it can cause some changes in terms of shifting on the other DNA pool for the other trophic level within the lake. We've tried to measure diversity of photosynthetic organisms that includes photoautotrophs and mixotrophs. So this particular photosynthetic organisms, we have noticed increase in their diversity. So which kind of supplement the idea that there is an increase in terrestrial input, increasing the nutrients in the water, and the nutrients in the water is is providing a very habitable condition for the phytoplankton to thrive in the water column. So you can observe increase in diversity within the DNA pool of phytoplankton and photosynthetic organisms. For parasites, we didn't notice any particular parasite that could harm the whitefish population. And in terms of diversity, they basically remain the same before and after 1950s. And for the consumers, that's basically including the whitefish, we've noticed a decline in diversity of consumer DNA pool after 1950s as in comparison to before 1950s period. And in terms of the interaction based on the models of the metabarcoding data that we've detected, before 1950s, most of the interactions are basically uh, comprised of interactions between terrestrial land plants, interactions between fungal spores that we've detected in the DNA. There are interactions of arthropods representing the zooplankton taxa present in the water. There's also craniata, which are basically the fish species and other mammal DNA that we've collected. So basically, it's highly limited to this group. But when we tried to reconstruct potential interaction network based on the DNA amplicons that we got from metabarcoding, become more diverse. So as you can see on the right side of the figure, a lot of nodes are basically coming from phytoplankton groups, indicating that after 1950s, phytoplankton become more dominant in terms of the abundance in DNA number, and at the same time in terms of how they interact with other taxa present within the lake. It still includes embryophyceae, which are terrestrial land plants, but there's becoming less. It also includes zooplankton interactions and mostly a diverse group of mixotrophs and phytoplankton groups that are capable of increasing primary productivity within the lake water column. So based on this data, I'd like to summarize the presentation first with the documented landslide in Copper Lake based on the indigenous knowledge is very useful information on how we can reconstruct and supplement this idea of the potential in reconstructing the history of Copper Lake. So the documented landslide in Copper Lake led to potential increase in sedimentation rate, as I've shown based on the sediment chemistry data. It leads to increase into it's caused by the increase in terrestrial input that might have caused the disappearance of whitefish. So basically, that's our existing hypothesis for the disappearance of whitefish species. And then changes in species composition of eukaryotic communities, the sedimentary DNA metabarcoding data, including the species targeted data based on the sedimentary DNA analysis, allows us to see shift in community composition within the DNA pool in Copper Lake through time. So it's basically a century worth of baseline, giving us an idea and an overview on the changes in the community composition within the lake and also around the lake, because it also covers terrestrial land plants, thus giving us a good retrospective tool to reconstruct lake history and assess potential impact of different anthropogenic activities and changes in land use within the area. And at the same time, weaving and braiding sedimentary DNA or eDNA approach together with indigenous knowledge could be a very, very useful tool for inferring history of a particular landscape, in particular, different lake ecosystems within the oil sands region. And after the summary, I really like this statement that Ave incorporated with one of the publications that we will come out with this work plan. Sedimentary DNA and indigenous knowledge can really complement each other. They are really good in augmenting each other in providing a more comprehensive narrative on the changes in history and community composition and diversity of different organisms within the lake. It gives you a very useful tool for predating human mediated changes within a lake ecosystem. Ave mentioned the theory about baselines and how difficult it is to determine a particular baseline and to do environmental impact assessment. But I think my favorite part, at least, which we find very useful for the community partners that we're working with, is that it gives them a piece of information that they can use to establish pre-development baseline that aid in the definition of setting a particular threshold so that they can support meaningful exercise of the Aboriginal and treaty rights within their territories. That's basically part of the year one of the work plan. So we've conducted 2 years of a project with the oil sands monitoring program. For the year one, we've collected, or Ave and the team collected sediment core from 6 lakes. And then for the year two, in collaboration with Keegan, they've collected sediment core from 5 more lakes. So this presentation only covers what we found out with Cowper Lake and the changes in Cowper Lake water quality and biodiversity history. But we have tons of data coming from other lakes within the oil sands region, and that's through the support of the oil sands monitoring program under the Indigenous community-based monitoring work plan. Yep, so that's the end of my presentation. Feel free to ask questions to me and to Ave.
Hey, thank you so much for sharing. I want to thank everyone for joining our science seminar today. Our next science seminar will be on Tuesday, March 11th, with Dr. Patrick Hannington presenting on spinning tails, exploring whirling disease in Alberta. If you are not on our mailing list and would like to be added, please contact the Office of the Chief Scientist at epa.ocs.gov.ab.ca. We'll end that for today. Have a great afternoon. Thanks again to our presenters for sharing their work with us and to everyone who tuned in. Thank you so much.